I'm Dr. Sarita. I'll be discussing with you the mesodermal derivatives during the embryonic period. Intra-embryonic mesodermal germ cell layer is important derivatives between the 4th week to 8th week of development, which is supposed to be the most crucial period of development. 4th week of development, the embryo is poppy seed and roughly about 7 millimeters in length. 8th week of development, the embryo is roughly kidney shaped or bean shaped. 8th week, the length of the embryo is roughly 2.5 centimeters or 1 inch long. 4th week, the development of umbilical cord and placenta starts working out. 6th week of development, the heart presents two important chambers and heart starts beating as early as 22nd day. 8th week, the ling buds enlarge or elongate and the fingers and toes are also forming in the fetus. Derivatives of intra-embryonic mesoderm germ cell layer and its present which is between the endoderm and ectoderm. Intra-embryonic mesoderm forms several distinct masses. The cells entering the primitive node becomes the notochord and prochordal plate. The first cells entering the primitive streak becomes the endoderm. Later, cells entering the primitive streak becomes the mesoderm, and cells that do not enter the primitive streak remain on the surface and become the ectoderm. Intraembryonic mesodermal cells are basically derived from the cells which are first derived from the primitive streak. These cells get detached from the primitive streak and they fill the space between the ectoderm and endoderm. Initially, the intraembryonic mesoderm forms a thin sheet of loosened woven tissue on the either side of the notochord. The mode of spread of intraembryonic mesoderm is cranially, which basically forms the cardiogenic mesoderm, that is the future heart. Caudally, it's known as caudal eminence, and this forms number of important pelvic structures. Laterally, intraembryonic mesoderm divides into three segments. Medially, somites, intermediate part is intermediate mesoderm, and laterally, lateral plate mesoderm. Intraembryonic mesoderm fills the space between ectoderm and endoderm except in the region of buccopharyngeal membrane, cloacal membrane and the median region which is occupied by the notochord. As neuralization starts, which starts as early as pre-somite stage, 15th to 21st day. Mesoderm also differentiates and subdivided into longitudinal grooves into three parts by the 17th day. Paraxial mesoderm medially, intermediate mesoderm in the middle, and lateral plate mesoderm laterally. Paraxial mesoderm medially, intermediate mesoderm in the middle, and lateral plate mesoderm laterally. Fate map is established during gastrulation. The cells that ingress in the cranial part of the primitive node becomes the prochordal plate or the buccopharyngeal membrane, and it also forms the notochord. Those cells at the lateral edges of the node and from the cranial end of the primitive streak becomes the paraxial mesoderm. Cells that migrate from the mid-streak region becomes the intermediate mesoderm. Those that migrate through the most caudal part of the primitive streak forms the lateral plate mesoderm. Paraxial mesoderm, which is the medial part of intraembryonic mesoderm, is a thick plate of tissue on the either side of the notochord. Lateral plate mesoderm. is a thin and extends up to the peripheral margin of the embryonic disc, and it is continuous with the extraembryonic mesoderm lining the amniotic cavity and the yolk sac cavity. Intermediate mesoderm initially connects the paraxial mesoderm with the lateral plate mesoderm. So therefore, to explain, the paraxial mesoderm is a longitudinal column of cells that lie on the either side of the notochord. Intermediate mesoderm, which gives rise to the urogenital system. Lateral plate mesoderm gives rise to body wall structures, and it is continuous with the extraembryonic mesoderm at the peripheral margin. Septum transversum forms the transverse bore of mesoderm at the cranial end, cranial to the buccopharyngeal membrane. It is only place where the mesoderm extends across the midline. This region later forms the future heart, diaphragm and liver. Caudal mesoderm lies caudal and lateral to the caudal part of primitive tree. In this region, it gives rise to the caudal structures including the pelvic viscera. So embryonic disc, initially flat and circular, gradually becomes elongated, broad in the cephalic region and narrow in the caudal region. The growth and elongation of the cephalic part of disc are caused by continuous migration of cells from the primitive streak region in the cephalic direction. The cephalic part, the germ cells begin their specific differentiation by the middle of third week, whereas in the caudal part, differentiation begins at the end of fourth week. As we see in gastrulation, where the formation of three germ cells continues in the caudal segments while the cranial structures are differentiating into number of tissues and organs later, causing the embryo to develop cephalocaudally. The paraxial mesoderm on the either side of the notochord. Intermediate mesoderm gives rise to urogenital system, and lateral plate mesoderm gives rise to the coelomic cavities and body structure, and it is continuous with the extraembryonic mesoderm. Mesoderm, initially a loose woven tissue, further differentiates into medially paraxial mesoderm which forms the somites. Intermediate mesoderm forms the urogenital system, and lateral plate mesoderm differentiates into two layers, parietal layer and visceral layer, and finally forms the coelomic cavity. So paraxial mesoderm showing the coronal section forming the somites. Intermediate mesoderm forms the nephrons, that is the urogenital system. Lateral plate mesoderm splits into two layers, the somatopleuric and splanchnopleuric. These two layers finally form the intraembryonic coelomic cavity, the pericardial cavity, the pleural cavity and peritoneal cavity. Coming to the fate of intraembryonic mesoderm. Fate of infraembryonic mesoderm, that is the fate of paraxial mesoderm, that is formation of somites and its various important structures. Fate of intermediate mesoderm, which finally forms the urogenital system. Lateral plate mesoderm, which forms the important body cavities. Fate of caudal mesoderm, that is a caudal eminence. Fate of transverse mesoderm, that is a cranial end, that is forming the cardiogenic plate, which forms the future heart. Now coming to the development of paraxial mesoderm, that is the most medial part of intraembryonic mesoderm. This medial part of intraembryonic mesoderm begins to organize into segments. Initially, solid cords of cell bilaterally located alongside of the notochord beneath the neural tube begins to organize into segments. Cranial segments are known as somatomeres and the caudal segments are known as somites. Somites and somatomeres forms the musculature of the skeletal, including the axial skeleton, body wall, muscles and structures related to head and neck. The first seven somatomeres, seven pairs of somatomeres, they form important structures related to head and neck region. Caudal somites, which are almost about 42 to 44 pairs, starting from the occipital base, they form a segmental organization of the body. Finally, to conclude that somites are segmental structures of the paraxial mesoderm, they are 42 to 44 pairs. Occipital somites, cervical, thoracic, lumbar, sacral, and coccygeal. First pair of somites appear around the 20th day, 3 pairs each day till 30th day. Then the rest of it is completed by 40th day, but in a slower rate. Somatomeres are also parts of paraxial mesoderm that is located in the head region. Somatomeres are first to 7 pairs. They form the mesoderm of the neck. They also take part in forming the striated muscles of the face, that is, the muscles which are derived from the second branchial arch, jaw, that is, the muscles of mastication which are derived from the first arch, muscles related to throat, that is, pharynx and larynx, that is, the constrictors of the larynx, intrinsic muscles of the larynx and constrictors of the pharynx, skeletal and muscles of the head and neck region basically related to the branchial arches. In addition, the somatomeres also gives rise to the extraocular muscles. Somites are caudal to the lower 7 somatomeres, and they are caudal to the otic vesicle. The first pair of somites appears in the occipital region of the embryo and proceeds craniocaudally or cephalocaudally direction. Somites appear between 20th to 30th day of development. The first pair of somites arise in the cephalic region of the embryo at the 20th day or the end of second week of development. From here onwards, the somites appear in a craniocaudal sequence at the rate of 3 pairs a day until the end of 30th day or the 5th week of intrauterine life. 42 to 44 pairs are present between 20th to 30th day of development. Cranial somites are the oldest and the caudal somites are the younger ones. Cranial four somites are occipital somites situated at the side of hindbrain. They help in formation of the skull base and muscles of the tongue, intrinsic and extrinsic muscles. Somites are 42 to 44 pairs. Occipital are 4, cervical are 8, thoracic are 12, lumbar are 5, sacral are 5 and coccygeal is 8 to 10 pairs. First occipital somite and last 6 to 8 coccygeal somites completely disappear. So remaining somites are approximately 34 pairs, which forms the axial skeleton, vertebral column and the base of skull. So after the first occipital somite and 7 coccygeal somites disappear from the original 44 somites, 3 occipital somites and remaining 31 pairs of somites which correspond to 31 pairs of spinal nerves. First 7 somatomeres give rise to the striated muscles of the branchial arches and extraocular muscles, including the skeletal structures related to head and neck region, that is, the skull bones, including the skull bones. 3 pairs of occipital somites form the base of the skull and muscles of the tongue. The rest 31 pairs of somites somites from the cervical region onwards give rise to the vertebral column. They subdivide into three components: sclerotome, which forms the vertebral column, myotome, which forms the muscles, and dermatome, which forms the dermis of the skin. So overall, to conclude, the somite period is between the 20th to 30th day of development, which forms 42 to 44 pairs of somites. So number of somites correlating with the age in days. 20 days or 20th day, we have 1 to 4 somite pairs. 30th day, we have about 34 to 35 pairs of somites. Around 25th day, we have, it corresponds to 20 somite period, and this is correlated with the closure of anterior neuropore. Around 28th day, the somite is 25 somites, and it corresponds to the closure of posterior neuropore. Correlating between the somites and the spinal nerve. Caudal to the occipital region, the number of somites are 31. 8 cervical, 12 thoracic, 5 lumbar, 5 sacral and 1 coccygeal. Somites here correspond to the same number of spinal nerves. Cranial somites. The 3 occipital somites are closely related to the hindbrain. Basically, they form the bones of the cranial base and muscles of the tongue, which is supplied by the 12th cranial nerve, that is hypoglossal nerve. Pre-occipital somites, that are somatomeres, which is related to the midbrain and also hindbrain, form extraocular muscles. These extraocular muscles are supplied by the 3rd, 4th and 6th cranial nerves, that is oculomotor, trochlear and abducens. Now coming to the structure of the somites, each somite is a mesodermal cells arranged in a concentric whorls around a central unit, donut shape around a small lumen. Each somite is divided into a ventromedial part, the sclerotome, and dorsolateral part, which is known as dermomyotome. So each somite, the differentiation is the ventromedial part is a sclerotome and dorsolateral part is known as dermomyotome. Schematic diagram showing the sclerotome, ventromedial part and dorsolateral part showing the dermatome and myotome. The dermomyotome is further divided into dermatome and myotome. Ventral and medial part of somites migrate around the notochord and the neural tube. The neural tube is a spinal cord. It is known as sclerotome and it forms the primitive vertebral column and the ribs. The remaining of the somite is the dermomyotome. The dermomyotome, the dorsolateral part of the somite. The cells here we find in the dorsomedial region and ventromedial region. These cells at these edges are known as myotomic cells. They form the muscle cells. Cells between these two groups of cells is the dermatome, which forms the dermis of the skin, which lies beneath the surface. Showing the schematic diagram of the somite, sclerotome,